So the interpupillary distance, IPD, is a distance in millimeters between the centers of the pupils' eyes. So figure shows how IPD is being measured here, okay? So this is how IPD is being measured, distance between the two pupils. So IPD is one example of a characteristic of human facial structure that shows variation. So you know that this variation, right, can be measured, and because it can be measured, it is quantitative, right? It's a quantitative variation, right? So let's look at the graph that's given. So it's a bar chart showing you y-axis is the number of people and x-axis is the interpupillary distance, so the phenotype, the phenotypic variation. Okay, y-axis is the number of people having it. So this is a normal distribution of the variation. So right in the center is the optimum phenotype. Okay, you learned already, right? So right in the center here, when there's a maximum number of people having the phenotype, this is the optimum phenotype of this population at this time. Optimum phenotype. Okay, so name the type of variation shown here. Now, before we name it, right, let's go back to look at what you have learned about variation. Have I started recording? Yes. Okay. What have you learned about variation here? Okay. So you learned that there are two types of variations in the phenotype. Either it is continuous or discontinuous variation. And it is represented by the two graphs in the bottom here. On the left side, you can see there's a very smooth curve, and the phenotype is actually quantitative. Okay, the phenotype is quantitative. And then on the right side, you can see that the phenotype is qualitative because we're talking about blood group. Blood group got blood group A, B, and O. So there's no value there. So we cannot have got a smooth continuous curve. So it's a discontinuous variation. Okay. So therefore, for the IPD that we showed just now is actually the x-axis is a value. So the value, even though they plotted the bar chart, right? Even though they plotted the bar chart, if you check the x-axis, they are all quantitative. The moment is quantitative, straight away it will be the continuous variation. So let me write here so that you know very well. This is a quantitative phenotype, phenotypic variation. And then the other one is a qualitative variation. Okay, so that straight away you know which is continuous, which is discontinuous. Quantitative is continuous, qualitative is discontinuous. All right, and then of course you must learn this up so that you can write answers for structure and essay question. What if the essay question come out? Okay, you want to be able to write the answers for all the possible essay questions, because essay question is a combination question. Combination means you got, okay, question 10, you got part 1, part 2. You find that, okay, this is part 1 question. And then part 2 question, you can answer very well. But suddenly part 1, you didn't study. Now how are you going to start? How are you going to write your answer? Then for the other essay question, part 1, part 2, also you're not very familiar. So therefore, you want to make sure that you can answer all the essay questions very well, okay, because you want to score A star, or you want to score as high as possible. So you want to do all the preparatory work well to make sure that you can score as high as possible. Okay, so do whatever you can do now to learn up everything you need to learn in the syllabus. Continuous variation. Okay, continuous variation is phenotype is quantitative. And then why is it so? What is the nature of the gene that leads to this continuous variation? When it's continuous, there are many genes that code for this phenotype. Many genes that code for this phenotype. And each of the gene, in this case, they code for the height. So therefore, different genes have the same that means all of them code for height. So they are the same. they code for height, okay? And when you have got all the genes together, it gives you an additive effect. That means each gene code for maybe 2x or 3x of the height. So when you got all the 5 genes together, you will add all the 2x, 3x, 4x, 5x together. So it gives you the additive effects to the height. So the genes code very similar phenotype, and the effect on the phenotype is additive. So this is continuous variation, okay? And of course, we know that genes got different alleles. For this case here, different alleles got very small differences among them. So got very small effects on the phenotypes. Okay. So when you got a large number of genes, so this is called polygenes, it means you got a lot of genes, they give you a combined effect on the phenotype. So this is for quantitative phenotype. The nature of the genes that code for it is this, like this. Okay, versus the discontinuous or discrete variation, where the phenotype is qualitative, like for example, ABO blood group or sickle cell anemia, right? They're all quantitative. So you can see that the moment the phenotype variation is discontinuous, every allele will code for a very different phenotype. You can see that, right? So if you got IAIO, you have got blood group A. When you got IB, you got blood group B. So there are different alleles will code for a very different phenotype. So therefore, different alleles will have got very large effect on the phenotype. Okay, we're talking about the alleles already. And then there are only one or a few genes that control this phenotype. Okay, like ABO blood group, we're talking about isohemagglutinogen, right? So the gene that codes for isohemagglutinogen. So this particular gene has got a few different kind of alleles, right? Versus this one here, you got many genes, whereas this one only got one gene. So when you study this one, you have to study with understanding. Yep. So different genes have got quite different effects on the phenotype. Doesn't look correct. Okay, yeah, different alleles got different effect. Different genes, of course, got even more different effects. Okay, if you're talking about different gene here. Okay, who's out? Okay, so taking opportunity to learn about this part well. Let's go back to the question. Okay, so name the type of variation shown in this one. Of course, this one is a continuous variation because the phenotype is quantitative. Okay, next, suggest and explain how genes and the environment, so keyword here, how the genes and the environment contribute to the variation in IPD in humans. Okay, yeah, so this one is your knowledge about VP is equals to VG plus VE, right? Remember, a variation in phenotype is due to variation in genotypes plus variation in environmental factors. So they're asking you to suggest and explain how these genes and environment contribute. So how does the gene contribute to the variation? So wait. So because this is a, let me type here first. Because this is a continuous variation. So you want to tell me about the property of the gene that code for continuous variation. Okay, so there are several or many genes, okay, that code for the IPD, okay, because it is a continuous variation. So you can have several or many genes. So you're talking about the genes now. What about it? Okay, next. So this gene have got additive effect, okay, or the genes interact to give an additive effect on the phenotype, which is actually IPD lah, okay, additive effect on the phenotype, which is IPD, okay. So this is how you apply your knowledge to answer the question. So this part is about the genes, how the genes contribute to variation. So how the genes interact to give the variation that you can see. So this is the property of the genes for continuous variation. So what about the environment? So the environment, because based on the equation they've given, you can write this equation. When you write your answer, you might get 1 mark for AVP, any other valid point, showing that you understand these 2 factors affect the phenotype. Okay, so environment has a big effect or significant effect on the phenotype. Okay, because the environment will affect the gene expression. Okay, so what do you mean by environment affects gene expression? So for example, this is similar to when you grow taller. So what are the nutrients that will support a person to grow taller? So here is what are the nutrients that will support the growth of the distance between the eyes, okay, the facial features. So it could be the nutrients. So you can say that the example, so what are the environmental factors that affects? It can be the diet. So what do they eat? It can be the age, okay, because you know, as a person grows older, the face will become broader. So it could be the age. Diseases will also affect this part. Chemicals, mutagens, okay, alcohol, okay. Now this one is all these things, right? The diet, the age, disease, it has to do with the mother, because the development occurs in the embryo, in the fetus. So how can it affect the IPD of this embryo or fetus when it's growing? Is to do with the diet, everything about the mother. Okay, the diet of the mother, age, disease, chemicals, mutagens, alcohol. Okay, in the utero. That means when the fetus is growing. Okay, so this one can affect the fetal embryo or fetal development. Okay, and presence of disease, presence of parasites as well, parasites in the uterus. Okay, so all these things you can think of. Okay, what are the factors which can affect growth? Anything that can affect growth, you think of that you can learn. Mutagens can cause mutation, right? Okay, so these are the possible answers.